Welcome, everyone. I'm Jody Siegel, here on behalf of the FDA CERSI, with Dr. Caleb Alexander on sabbatical. I have the honor of leading the CERSI in his absence, so I'm delighted to be able to welcome this month's speaker. This month, we have Dr. Elsa Anagnostou, who's an associate professor of oncology and director of the Thoracic Oncology Biorepository and co-leader of the Molecular Tumor Board here in the Sidney Kimmel Cancer Center, Johns Hopkins. She graduated from medical school of the National and Kapodistrian University of Athens, Greece, and received a PhD from there as well. She completed her internal medicine residency at Yale New Haven Hospital and then trained in medical oncology at Johns Hopkins. She's a translational cancer investigator focused on large-scale genomic, multiomic, and liquid biopsy analyses in human cancers. She focuses particularly on understanding the molecular mechanisms of response and resistance to immunotherapies and translating this into novel technologies and therapeutic approaches. She wants to transform medical oncology to molecular oncology, where treatment decisions are tailored not only by the baseline genotype, but informed by real-time information from liquid biopsies. And that is her talk today, and I'm so excited to hear. So please, cue.
Great, thank you, Dr. Siegel. Good afternoon, everyone. Thank you again for the great introduction. It's a great pleasure to present at the FDA CERSI lecture series. And today I'm going to be discussing the evolving field of liquid biopsies, particularly particularly the efforts to enhance translation from bench to bedside and enable precision oncology. Here's my disclosures. Now, liquid biopsies have been emerging as potentially powerful, non-invasive means of detecting tumor burden and tracking tumor evolution. And of course, the clinical utility of liquid biopsies is supported by an ever-growing number of studies showing that circulating cell-free tumor DNA, or we call it ctDNA, and ctDNA dynamics can be implemented in early cancer detection, detection of minimal residual disease, and monitoring of therapeutic response. One of the challenges with liquid biopsies, though, is inherent to the scarcity of the analyte, which is the tumor-derived mutant fraction of cell-free DNA, again, or ctDNA. And the mutant tumor sequences are present in very small quantities relative to normal DNA fragments. Just to give you an example, a 10 ml blood draw yields about 4 to 5 ml of plasma, 20 nanograms of cell-free DNA, that's in a healthy individual. And that equates to about 6,000 genome copies. And now if you look at the bottom of the figure here, you can appreciate the challenge with detection of very, very small levels of ctDNA, as there may be less than 10 or even less than 1 tumor genome copies in our plasma sample, right? So conventional PCR and sequencing error rates are very similar to the levels of mutant DNA. Now, over the past decade, we've certainly seen the development of several targeted next-generation sequencing technologies for detection of mutations in ctDNA. These employ deep and redundant sequencing together with implementation of nucleotide barcodes, size selection of the genomic library, sequencing error suppression algorithms. These are bioinformatic approaches to boost assay sensitivity by improving the signal-to-noise ratio. And altogether, these steps enable sensitive and specific detection of mutant sequences from cell-free DNA. And what I'm showing here is two representative examples. On the left is a hybrid capture-based approach where genomic regions of interest are captured. This is the basis of several commercially available assays like Guardant, Foundation, and others. And on the right, I'm showing multiplex PCR NGS approaches that are the basis of a little bit more sensitive commercially available assays like Natera's Signatera. And you can appreciate, though, by both approaches that the error rate with these, which is shown for one of these methods in the blue line here, is several orders of magnitude lower compared to conventional NGS, which we're showing here in the red line. Now, we discussed briefly technical noise and technical advancements to boost signal-to-noise ratio in liquid biopsies, but I would like to take a minute and discuss biological noise in liquid biopsies. Now, it's important to realize that the majority of cell-free DNA in plasma is not released by tumor cells. It's actually released from hematopoietic cells. And therefore, any blood-based NGS approach has the potential to detect clonal hematopoiesis-related mutations, and these may be misclassified as tumor-specific. How prevalent is this? It's actually quite frequent. Overall, about 50% of cell-free DNA mutations can be of clonal hematopoiesis or germline in origin. And these, to make it even more complex, these cell-free DNA mutations from white blood cells, these can fall in genes that are not canonically associated with clonal hematopoiesis. These are not just mutations that fall in DNMT3A or TET2, for example. Again, highlighting the importance of developing ways to carefully characterize the origin of mutations detected in liquid biopsies prior to making any clinical decision based on liquid biopsies. Now, having briefly discussed the capabilities and limitations of liquid biopsies, I wanted to focus on the clinical implementation of these approaches in measuring residual disease and tumor burden in several clinical scenarios. Now, ctDNA can be tracked longitudinally for minimal residual disease, or MRD, as I'm going to be referring to, in the early stage paradigm that I'm showing here on the left, as well as in the context of response monitoring in the metastatic paradigm that I'm showing on the right. And I will discuss the MRD setting first. Now, there is a number of studies that support the notion that MRD, ctDNA positive, is a poor prognostic marker. And the point that I want to make here is that the clinical sensitivity of MRD, which is the percentage of patients who relapsed and who were ctDNA positive, this is actually higher when longitudinal MRD assessment is implemented compared to single or snapshot MRD analysis, something that we call landmark MRD, as you can appreciate here. And this holds true, actually, if you look in D here, across ctDNA detection platforms. Therefore, one thing to consider is the longitudinal monitoring may increase the probability of ctDNA detection and actually distinguish true MRD ctDNA positive cases. Now, we've learned some important lessons from several clinical trials that have now been reported, and I'm going to review several, starting with the neoadjuvant clinical trial Checkmate 816. We learned that patients with early stage non-small cell lung cancer that received neoadjuvant nivolumab and chemotherapy, patients that showed ctDNA clearance preoperatively, these had a longer event-free survival and were more likely to attain a pathological complete response at the time of resection. And from from the IMvigor10 clinical trial, we learned that patients with operable urothelial cancer now that showed ctDNA persistence post-operatively, this is a landmark MRD assessment. These had improved disease-free survival and overall survival when they received atezolizumab compared to observation. Now, how about ctDNA clearance at the MRD time point? And what I'm reviewing here is findings from the dynamic clinical trial, where a ctDNA positive result at 4 to 7 weeks after surgery informed adjuvant chemotherapy with an oxaliplatin or 5-FU regimen for patients with stage 2 colon cancer. And in the same trial, patients who were ctDNA negative at this MRD assessment were not treated. Of course, this was a positive trial reported not too long ago in New England Journal of Medicine, which was good news for ctDNA approaches. But what I wanted to highlight from this trial is the fact that in the ctDNA guided group, we still had recurrences. We saw recurrences occurring in about 6% or 15 out of 246 patients in that trial. And these of patients that were actually ctDNA negative. Therefore, the point that I want to make here is that it is important to realize that a fraction of MRD ctDNA negative patients will, especially if MRD is determined at a single time point, these may still experience disease recurrence despite the use of a tumor-informed sensitive assay, as was done in this particular trial. This is where I think clinical trials stratifying MRD ctDNA positive patients are going to be informative as ctDNA technologies continue to evolve. And this is from a recent JCO review outlining the escalation or de-escalation trial design in the early stage disease setting based on landmark or longitudinal ctDNA MRD detection. And one point I want to make here is related to de-escalation. While conceptually it makes sense to de-escalate based on ctDNA negative, we have much less data from retrospective cohorts to inform when and how to de-escalate therapy. Now, with respect to regulatory decision-making, certainly we need further clinical validation of ctDNA MRD, especially in the context of clinical trials. ctDNA MRD after definitive therapy can be used as a stratification factor in studies enrolling patients both negative and positive for ctDNA, or in separate trials for ctDNA MRD positive and ctDNA MRD negative cohorts, and these can be considered separately. Now, I wanted to switch gears to the metastatic setting, again, focus our attention to the right-hand side of the graph, and focus on residual disease monitoring during immune checkpoint blockade for patients with metastatic cancer, especially non-small cell lung cancer, which is what we are studying. Liquid biopsies are certainly gaining momentum in immuno-oncology as well, and these can be utilized to rapidly and accurately determine clinical responses. Optimal patient selection for immunotherapy is a critical challenge, and it is truly intensified by the heterogeneity of clinical responses that we observe and inefficiency of imaging to fully and timely capture therapeutic response. Typically in immuno-oncology, we use snapshot biomarkers to guide therapy selection, such as PD-L1 expression and tumor mutation burden, but these come with their own challenges. And together, all these challenges truly highlight the urgent need to develop molecularly informed strategies to enable early and accurate response assessment and facilitate long-term response monitoring during immunotherapy. And here I summarize the approaches for the detection and analysis of ctDNA in the context of immuno-oncology. These follow the same paradigm as with MRD detection that we previously discussed. In the context of immunotherapy, serial sampling and longitudinal ctDNA analysis can capture the evolving tumor burden as well as emerging mechanisms of therapeutic resistance. And ctDNA analysis in the context of immuno-oncology may provide, may be particularly helpful in studying difficult to evaluate tumor types, for example, mesothelioma, or assist with patient selection for immunotherapy and subsequent response monitoring. We're going to talk more about this, particularly in radiographically stable disease, and we're going to talk more about this as well in the coming slides. Overall, accurate therapeutic response monitoring by liquid biopsies can inform decisions also on the optimal duration of immunotherapy and appropriate strategies for treatment escalation or de-escalation based on ctDNA molecular responses. Now, liquid biopsies may complement tissue biomarkers in predicting therapeutic response in the context of immunotherapy, especially when tissue biomarkers fail to predict outcome. I'm going to give you an example using tumor mutation burden that is very commonly used as a predictive biomarker for immunotherapy. This is certainly exemplified in the setting of hypermutated tumors in the context of mismatch repair deficiency. However, outside of mismatch repair deficiency, what we see repeatedly is that TMB is an imperfect biomarker of response, and there's inherent challenges that have to do with technical limitations, such as lack of standardization and harmonization, a universal threshold. And the point that I want to make here is the following: tissue TMB or tumor mutation burden estimates are subject to sampling bias and are strongly affected by low tumor purity. Very simple concept, however, very much underestimated in the clinical setting. Now, in order to determine how tumor purity may affect TMB, what I'm showing you here is simulation analysis to evaluate the tumor purity needed to accurately determine TMB in the setting of different clonal compositions. And as you can appreciate from the in silico dilution experiment shown here for a TMB high clonally complex tumor that I'm showing in the top row, and a TMB low clonally homogeneous tumor, just 2 clonal clusters that we're showing here in the bottom row, the power of detection of mutations dramatically decreases with decreasing tumor purity. And what you can appreciate is that the lower spectrum of tumor purity, I'm just showing 40% as an example here, TMBs of clonally heterogeneous TMB high and clonally homogeneous TMB low tumors, these become the same. They're indiscernible, again, highlighting the challenges with using tissue TMB as a predictive biomarker. Now, could blood-derived TMB remedy this issue? Of course, ctDNA, the ctDNA pool is representative of mutations originating from distinct primary and metastatic sites and conceptually better captures tumor clonal heterogeneity. And as such, liquid biopsies may offer greater accuracy for TMB determination compared to liquid biopsies. Certainly, blood-based TMB has been explored as a predictive biomarker of immunotherapy response with encouraging results in retrospective assessments. I mean, these were the popular OAK, MYSTIC trials in non-small cell lung cancer. However, what we saw was that when prospective analysis were implemented using pre-specified blood TMB thresholds, these studies, and I'm showing 2 here, BFIRST and BFAST, these did not fully confirm the predictive value of blood tumor mutation burden. So in the phase 2 BFIRST trial, this was first-line atezolizumab in patients with locally advanced or metastatic non-small cell lung cancer, a high TMB of higher than 16, blood TMB that is, higher than 16 mutations per megabase, that was associated with a higher overall response rate. However, these findings did not translate into a significant difference in progression-free or overall survival. And similarly, in the BFAST study that I'm showing on the right, that again, similarly prospectively assessed blood TMB as a predictive biomarker of immunotherapy response, there was certainly a trend towards a longer progression-free survival in patients with blood TMB more than 16 mutations per megabase that again received atezolizumab, but there were overall no survival benefits as you see here in the bottom right. And certainly there are several technical and biological limitations that have to be considered and have to do with lack of algorithm standardization, threshold harmonization for blood tumor mutation burden, and also accurate exclusion probably of alterations that may be associated with clonal hematopoiesis that we previously discussed. Now, in my opinion, dynamic ctDNA assessment, that represents the most promising biomarker in the context of immunotherapy response for patients with metastatic disease. We and others have applied ultrasensitive next-generation sequencing methods to analyze ctDNA in serial blood samples from lung cancer patients during treatment with immune checkpoint blockade. And collectively, what these analyses have shown is distinct patterns of ctDNA-based molecular responses that are actually reflective of patients' clinical outcomes. So patients with a ctDNA molecular response, as I'm showing here in the upper left, have a dramatic reduction of ctDNA down to undetectable levels. This is reflective of long-term clinical benefit. And in contrast, for patients with primary resistance to checkpoint blockade, ctDNA levels have either limited fluctuations or display a rise, as we're showing here, after therapy initiation. Now, I'm going to go back to a determination of cellular origin for mutations detected in plasma NGS. This is important. Accurate determination of variant cellular origin, it is key in assessing ctDNA dynamics and molecular response. So as a reminder, a good fraction of alterations of mutations detected in plasma NGS could be clonal hematopoiesis related. And one way to overcome this issue and accurately assign cellular origin of variants would be to perform matched leukocyte DNA sequencing to practically filter out clonal hematopoiesis variants. This approach allows for accurate classifications of plasma variants in clonal hematopoiesis and tumor-specific categories, as in the case that I'm showing here, and that in turn allows for assessment of ctDNA dynamics and molecular response. And you can appreciate if one looks. The case that I'm showing here, this is a patient that actually derived clinical benefit from first-line immune checkpoint blockade. If you look at all alterations in their trends, cannot really make sense in terms of ctDNA dynamics. But then when we accurately classify variants based on cellular origin, one can make the determination of ctDNA clearance or molecular response, which actually reflected the long-term outcome of this patient. Now, employing a matched leukocyte DNA plasma approach, changes in levels now of tumor-derived variants that you can see here on the right, of course, not germline or clonal hematopoiesis-derived variants. This is what is predictive of durable clinical benefit to immune checkpoint blockade. Here we're showing for patients receiving first-line immunotherapy. And again, employing a matched leukocyte DNA plasma approach, the quantitative variation in ctDNA levels is linked with long-term outcomes, such as patients with ctDNA molecular response that we're showing here in the yellow survival curves. These have a longer progression-free as well as overall survival with immunotherapy. And of course, the association between ctDNA response and improved survival, this is further evidenced by a pooled analysis in the CT Monitor project from Friends of Cancer Research. In this initiative, ctDNA kinetics from 5 smaller immunotherapy-treated non-small cell lung cancer cohorts, these were harmonized, and the association between ctDNA dynamics and long-term outcomes was assessed in the larger pooled data set. Despite a significant cohort and methodology heterogeneity, we did observe there as well that there was a significant association between ctDNA reduction and better outcomes, or ctDNA persistence and worse outcomes that we're showing here with respect to OS, PFS, and overall response rate. Now, this association of ctDNA response with IO response, it has been reported in several proof of concept studies. But what I want to emphasize is the fact that there are several outstanding questions that I'm going to pose sequentially. The first one has to do with the definition of ctDNA molecular response itself. And I would argue that the definition of molecular response, be it, for example, reduction of maximal variant allele frequency by 50% or complete elimination of ctDNA, this can be ctDNA assay specific. It may relate to the LOD of the assay. And what I'm showing here is variation in mutant allele frequency replicates. This is from tumor-specific mutations from a mixture of DNA from tumor cell lines that are spiked into wild-type DNA. These are dilutions from 100% down to 0.1%. And what I want you to focus on is that the coefficient of variation in this MAF estimate, mutant allele frequency estimate, increasing obviously with decreasing mutant allele frequency, and importantly, the relative percentage uncertainty in the MAF estimate. Now, all that these analyses say is the following: take a MAF of 1%. For example, a mutant allele frequency of 1%, which is right here where the dotted line is. The 95% uncertainty in that estimate, in that level of mutant DNA, ranges from -50 to plus 50%, right? So the true MAF can be anything from 0.5 to 1.5%. Thus, a greater relative reduction is required to determine ctDNA response for lower maximal mutant allele values, and that's where complete ctDNA clearance may come in. Now, another question that has not been definitively answered is the optimal time point to determine ctDNA response. In the IO space, we know that this is somewhere between 4 to 9 weeks, with reliable ctDNA responses actually seen as early as after 1 cycle of immune checkpoint blockade. It is important to also consider the treatment modality. I'm showing you a representative example here where you can appreciate the faster ctDNA clearance with chemoimmunotherapy compared to immunotherapy alone that we're showing on the right. That makes sense given the cytoreductive role of chemotherapy, but certainly what we need, and I hope I emphasized enough, is harmonized definitions of ctDNA thresholds, harmonized definition of molecular response, as well as pinpointing the optimal timing for assessment of ctDNA responses. So how do we do this? How can we gain more granularity, answer these questions, and move from these proof of concept or low resolution studies, into higher resolution? And I think the answer here is to establish the value of ctDNA as an early endpoint of immunotherapy response in clinical trials. One scenario is shown here. This is a trial led by the Canadian Cancer Trials Group. And the question we're asking is, which treatment-naive PD-L1 positive metastatic non-small cell lung cancer patients can be treated with pembrolizumab alone versus pembrolizumab and chemotherapy. And this trial is conducted in 2 stages, a lead-in initial stage that I'm showing here on the left. This is now completed. And we're addressing the questions that are rather rhetorically posed before, which are the following. What is the definition of molecular response? When does it happen? And what is its concordance with radiographic response? We need to answer these questions before we move to clinical trials. Now, the results of this clinical trial will be presented in an upcoming meeting, but I wanted to give you a flavor of the findings as well as the challenging questions as we move to the interventional stage of the trial. Again, here, molecular response was very distinct from molecular disease progression, and ctDNA kinetics can certainly identify patients with molecular disease progression that can be subsequently randomized to intensified therapy, while patients with molecular response can be spared. Now, what is key is understanding the true concordance between molecular and radiographic responses, and how do these actually capture long-term outcomes. And this can only be done in the clinical trial setting to get to answer the question in terms of how do we handle patients that fall in the discordant groups that I'm showing here, the ctDNA responders but resist non-responders, or ctDNA progressors but resist responders. Now, related to that, are there specific patient subsets that ctDNA can be particularly informative? And can ctDNA be informative beyond radiographic response assessments? So what I wanted to take a minute here and discuss the role of ctDNA to better characterize radiographic stable disease, which, as we know, is a heterogeneous entity, especially in the context of immunotherapy. And this is a preliminary proof of concept analysis from a smaller analysis that we did several years back from patients treated with immune checkpoint blockade that were all stable by imaging. And what you can see here is that for this group, ctDNA response predicted outcomes such that patients they all had stable disease, right? But patients that cleared ctDNA had significantly longer progression-free survival compared to those that did not clear. Now, all that these findings suggest is that radiographic imaging in this particular subset may not be as effective in terms of detecting the magnitude of therapeutic response, and that ctDNA response may be of particular value in assessing therapeutic response in this setting. Now, how would a ctDNA-informed interventional clinical trial look like in the immunotherapy space? A clinical trial schema is shown is shown here, and I want you now to focus on the right-hand side. This is again BR36, our 2-stage phase 2 trial of molecular response adaptive immunochemotherapy trial for patients with metastatic non-small cell lung cancer. Now, here, patients with ctDNA molecular disease progression, these are those ones down here, these can be via the approaches that I just described can be accurately and rapidly identified and subsequently randomized to either continue on pembrolizumab versus treatment intensification with addition of chemotherapy to the pembrolizumab backbone. This is one scenario. There is several additional clinical trial schemas that one can think about in terms of implementing ctDNA-informed clinical decision-making. Now, dynamic profiling of ctDNA past the first assessment of molecular response is informative and can reveal additional ctDNA patterns that are reflective of tumor evolution, as shown here. Sustained ctDNA elimination that signifies typically ongoing response, long-term clinical benefit. But there's additional patterns, and we are able to detect those by longitudinally tracking ctDNA. There is ctDNA recrudescence that we're showing here. This refers to variants previously cleared and now re-emerging, or ctDNA emergence, and this refers to emergence of new variants. And these provide more insights in clonal evolution during therapy. Now, in addition to the quick fail approach, if you will, that I described previously and showed the BR36 clinical trial as an example, as we gain more insights into the longitudinal clonal dynamics, we can implement ctDNA in treatment de-escalation, as I'm showing here in the upper left, in the setting of sustained ctDNA clearance, as well as early switch. I'm showing in the upper right and bottom left in the setting of ctDNA recrudescence or emergence, and the latter may also have a role in refining drug development strategies and establishing ctDNA as an early endpoint to support drug approvals. Now, I wanted to very briefly discuss emerging approaches leveraging plasma-targeted next-generation sequencing or whole genome sequencing for precision immuno-oncology. Targeted NGS data can be leveraged to derive copy number profiles in cell-free DNA, estimate what we call plasma aneuploidy that can be tracked over time, similar to how we're tracking mutation levels. And I'm showing you such an approach here, where we're showing two examples of plasma aneuploidy clearance. This is shown on top, and persistence that is shown here at the bottom. Plasma-derived copy number gains are shown in red, and copy number losses are shown in blue. So you can appreciate the shrinkage of plasma aneuploidy to euploid state, like it all becomes gray, in a patient that is clearing ctDNA and responding, and tumors regressing with immunotherapy. And in contrast, or you can compare this with persistence of plasma aneuploidy that in a patient with primer resistance. And what makes this approach exciting is that tracking plasma aneuploidy may complement mutation-based molecular response assessments. And I'm showing you here 3 examples of patients with undetectable mutations for which we were able to make a determination of molecular response based on aneuploidy dynamics. And the integrating ctDNA features, copy number and sequence-based, can augment the sensitivity of plasma targeted NGS, as we're showing on the right here. We can appreciate the AUC of the integrated or the combined approach is actually better. Now, taking it a step further, an important consideration overall is that we may be reaching a plateau in sensitivity of mutation-based liquid biopsy approaches. And, these are expensive assays. So, to what extent can these be widespread, universally used is questionable. So rather than going after mutations, we have shown that detection of unique patterns in DNA fragmentation, this may differentiate cancer from healthy individuals. And such an example is shown here, where practically cell-free DNA fragmentation profiles are determined across the genome, and machine learning is employed to characterize whether individuals have cancer or not. This is graphically represented on the left. And while this approach was developed for cancer early detection, subsequent survival analysis showed that changes in fragmentation patterns after therapy, these may predict outcome. What I'm showing you here on the right is for a subset of metastatic lung cancer patients treated with targeted therapies. So certainly we don't have a lot of evidence as of yet, but we will be seeing more efforts of these broader genome-wide approaches in the IO response monitoring space in the very near future. Now, all the liquid biopsy efforts that I described are focused on bridging bench research with clinical oncology and enable the earliest best decisions for our patients with precision. So certainly the genomic complexity of tumors that we capture with liquid biopsies has certainly allowed for this, but we have a long way to go. So kind of bridging to the more clinical part of my talk, there is an expanding list of genomic alterations that are linked with FDA-approved therapies on the basis of improvement of clinical outcomes. But we constantly face this question as how do we match unvetted, what I call unvetted genomic alterations, mutations that we don't know a lot about that are detected in liquid biopsies. How do we pair all of these with effective treatment pairs? Can we just do this by going through what is included in commercial NGS reports in terms of mutation characterization now? But unfortunately, that's not the case. Variant annotations can be generic, can provide very limited information. They don't typically, there's no differentiation between tumor-derived and clonal hematopoiesis mutations. We talked about this. So what happens is that clinicians, oncologists typically face a vast volume and variety of molecular data that can be processed very quickly. But this is done to support vital clinical decisions. And I want to make the point here that while there's certainly significant advances in diagnostics for tumor profiling by liquid biopsies, a solid analytics backbone that supports the practical implementation within and across healthcare systems, this is not as developed. And certainly the delivery of precision medicine is disrupted to an extent by this paradoxical paucity of analytical tools to match genomic targets that are identified, say, in liquid biopsies with molecularly driven therapies. And that widens the gap between an exponentially increasing drug development field and the actual benefits for our patients with cancer. Now, there's certainly a plethora of resources that can assist with mutation interpretation, but this is a loaded slide. There's certainly several resources that one can use, but the currently available resources may be limited by, for the most part, manually performed, complex, time-consuming, error-prone gene queries, and ultimately lack the information needed for prioritizing emerging therapies in a scalable manner. And important to note that these remain out of reach for the vast majority of community physicians, right? So we certainly have knowledge bases, meta-knowledge bases, but how do we actually, how do we synthesize all the data? So in my opinion, this represents a challenge that has limited the broad application of precision oncology and contributes to the overall low participation rate in cancer clinical trials. And of course, these challenges are only amplified outside of the academic setting among community oncologists. Now, unique to academic institutions, molecular tumor boards, these are multidisciplinary expert opinion-based precision oncology teams. These can assist with this matching of genotypes with molecularly driven clinical trials. However, these initiatives are largely academic institution specific and poorly non-scalable efforts. So how do I address this? How do I address this unmet need? So I wanted to spend the last 5 minutes of this lecture to discuss an ongoing effort under the FDA Hopkins CERSI that myself and Taxiarchis Boutsis are leading. Our primary objective here is to develop an end-to-end precision oncology analytical tool that can enable clinical decision-making and patient selection for genotype-driven clinical trials. And very briefly, this effort utilizes open source tools, retrieves data from public repositories, processes multiple source and vendor sequencing files, and leverages a common data model that I'm going to be talking to you about. And the premise of this work is that it can enhance the implementation of precision oncology, even in institutions that do not have access to, say, in-house expertise, in clinical genomics. Now, in preparation for such programmatic clinical genomics data integration, we developed a precision oncology common data model, or core data model rather. This is a superset of the ASCO minimal common oncology data elements, or MCODE model. And this allows for harmonized clinical data pools across healthcare systems and institutions. Most of these data elements are mapped to elements, to FHIR elements, and this truly allows for harmonized clinical data pools across institutions. Now, this is very dense. What I'm showing you here is very dense, but you can appreciate the elements of the core model that are projected here, which are organized in modules related to the patient, the disease, the tumor sample analyzed, the NGS assay, the NGS output, clinical outcomes, and then incorporation of precision oncology expert recommendations. Now, these initiatives on automatic liquid biopsy mutation annotation, characterization, matching with clinical trials, as well as the clinical genomics data integration that I showed you, these can be leveraged in the context of precision oncology clinical trials. And I would like to show you such an example. This is projected here. This is a trial that was recently activated here at Johns Hopkins, where we're using liquid biopsies to match genotypes from plasma, next-generation sequencing from liquid biopsies, with molecularly driven clinical trials, with the primary objective to validate the feasibility of the approach in informing clinical decision-making and in validating the clinical utility of longitudinal liquid biopsies. Now, wanted to end by summarizing a very, very dense topic. I hope I gave you a good flavor of the field today. So tumor-informed or tumor-agnostic liquid biopsy approaches, these are tested in a variety of clinical diagnostic applications in precision oncology. There's ultrasensitive NGS approaches. We certainly need to do better, but these focus on MRD detection. These, in my opinion, the technology is still evolving. And needs to evolve prior to incorporation into clinical trials. And then in the metastatic setting, ctDNA kinetics, I think we're there. ctDNA molecular response, it truly represents a promising early endpoint of therapeutic response. And I think we're ready to test in clinical trials. And in doing all this, it's important to select the liquid biopsy approach based on its diagnostic application and intended use, and ensure that biologically relevant alterations are assessed. The technical performance is what it should be, and in order to minimize both technical and biological error that we discussed today. And certainly we need additional studies in the context of clinical trials to further support the implementation of liquid biopsies in clinical cancer care. So I'm going to stop here to allow for a few minutes of discussion, but I wanted to acknowledge the great work of folks in my lab right here in the thoracic oncology group, within the molecular tumor board at Johns Hopkins, and our collaborators and funding sources, especially through the FDA CERSI mechanism that supports several of the efforts that we discussed today. And as a final note, I wanted to highlight a 10-article series on liquid biopsies, from cell-free DNA biology to applications and regulatory implications, that Mark Stewart and Friends of Cancer Research, and myself, we put together for the Journal of Immunotherapy for Cancer. I wanted to invite you to take a look at the collection. You can just scan the QR code here, get to the landing page. So I'm going to end here, and happy to open it up for discussion.
Good. Thank you, Dr. Anagnostou. Fascinating and really cutting edge. So perfect for today's series. I will begin, and we will gather questions in the Q&A, which I'll read. So thank you, everyone, for your questions. You touched on it a little bit, but could you say more about the regulatory pathway for this? What is FDA requiring?
Yeah, I would argue that the evidentiary road truly depends on the setting, right? And I hope I showed the kind of two different clinical settings, Elise, right? So when we're talking about minimal residual disease detection, which is certainly a field that liquid biopsies can be very informative, we need more. We need to see more in terms of the technology. We need to develop additional ultra-sensitive approaches to detect those very, very small quantities of tumor-derived DNA in the circulation. And then in terms of implementing ctDNA analysis and decision-making into clinical trials, one can certainly randomize in terms of after definitive therapy, after surgery, let's say, can randomize based on ctDNA MRD status, positive or negative, for adjuvant therapy. Now, in the metastatic setting, I think we're a little bit closer. What we see in the metastatic setting, of course, there's more circulating. There's a higher circulating tumor burden, right, that we capture, I think, quite adequately with the technologies that we currently have. In lung cancer, to give you an example, it's about 20% that we don't detect, right, with the current technologies. And so in the metastatic setting, what we truly can, I think everyone or almost everyone in the field agrees upon is determination of molecular disease progression, right? So these, patients that do not clear, or ctDNA is not decreasing, and these are the patients that we can intervene upon. And the evidentiary road there includes precisely putting this clinical trial, such as the one that I showed you, which have to do with randomization of such patients to either treatment intensification or staying on the prior therapy, and then measure out. There's a lot of value, and I think we need to do this in the context of clinical trials in terms of actually thinking about randomizing all discrepant cases, right? It's just that we haven't done that many systematic analysis to be able to do, meaningful analysis of the discrepant molecular responses with RECIST responses.
I have a follow-up question, but that's not fair. Okay, I'm going to go to the chat. So we have a question. Do you think liquid biopsy will replace traditional tissue biopsy?
Yeah, that's a great question. And certainly liquid biopsies can complement tissue biopsies, right? Especially when a tumor tissue cannot be obtained because it's in the context of metastatic disease, where there's biopsy failures or whatnot. But one important point here that I want to make is that, and this is what the FDA's guidance is as well, which has to do with when ctDNA or liquid biopsy analysis are negative, right? So this is accompanied by reflex testing of tissue if tissue is available. So it's important to recognize that liquid biopsies also have, they have limitations in terms of their sensitivity with the current assays that we're using. And so I wouldn't necessarily argue that liquid biopsies are going to replace tumor biopsies and tumor NGS. I would look at these two modalities in a complementary manner. There's a lot that we learn from tumor tissue next generation sequencing that you can imagine one could combine with longitudinal assessments, kind of real-time assessments of circulating tumor burden, which is a unique feature of liquid biopsies, right?
Great. So the next question may be a little outside of the scope of what you want to address, but would a more developed health information exchange across health systems improve the challenge in oncology precision medicine?
Yeah, absolutely. And I think, I started talking about harmonization and standardization of clinical data capture within precision oncology initiatives. This is key, and I think what we're seeing is this great heterogeneity in terms of how information is captured and interpreted within different health systems, right? So we truly need these types of initiatives, and there are several actually that are ongoing right now to harmonize data capture, whatever data modality these may be, right? So all health-related data, and also implement ways to communicate and learn from each other. So I think that's an important point overall for precision oncology.
Mm-hmm. Right, and just the rest of the question was about statewide limitations, and I don't really think there are. I think it's HIPAA protected data and falls under the regulations largely.
Yeah, I agree.
All right, if I could go back to my follow-up question. It was interesting when you say the minimal residual disease. So you really think FDA won't require just demonstration against existing imaging? They will require trials for that, for hard clinical outcomes?
Right, but of course, one of, I think the FDA's guidance is more studies and, more in meta-analysis, and pooling of previous studies to understand the signal. But truly, I think, and we have started seeing signals, right, in MRD trials that have already resulted, and we discussed some of them. Truly, we need to see how well does, we need to understand the clinical sensitivity of the MRD assessment, right? And so whether it's landmark MRD or longitudinal MRD, I think these things need to be established. But truly, I think the gold standard would be trying to capture recurrence, which of course is radiographically determined, and long-term outcomes.
Right. Oh, you don't need to convince me, but is that what's going to be needed to convince FDA?
I think so. I think clinical, readout of additional clinical trials is hopefully going to convince the FDA. I do think that in the MRD setting, we do need to do better with respect to technology, though. And the sensitivity of the assays that we have in our hands.
Mm-hmm. Good. And another question, this person's interested if certain liquid biopsy assays are able to accurately differentiate between somatic versus CH, not sure what that stands for, or would a follow-up clinical evaluation still be necessary to confirm variants?
That is a great question. And so plasma only analysis, if you do, next generation sequencing from a plasma sample, and then there is a number of variants detected, a number of mutations detected, we cannot tell what the cellular origin of the variant is unless at the same time we know what is the tumor genetic landscape by matched analysis of tumor tissue and matched analysis of white blood cell DNA to get a sense of what is clonal hematopoiesis related. Now we can make, inferences if we see mutations, for example, in DNMT3A or TET2, these are prototypically mutated in clonal hematopoiesis. But again, this is not definitive. And I can tell you, from reviewing liquid biopsies constantly in the context of the molecular tumor board at Johns Hopkins, when, what is causing us headache is true mutations in P53. Well, P53 is mutated in most solid tumors as well as in clonal hematopoiesis. So, we see these mutations, their levels are at, they're at exactly the same level or very similar. We cannot tell the difference. So that's why it is truly important, especially when one wants to look at ctDNA dynamics, molecular response, right, make a therapeutic decision. It is truly important to filter out these mutations. Now, how can one do this, right? So again, matched white blood cell DNA sequencing, this is not clinically offered right now. right? But we, at least I know, I can tell you, my group is working on machine learning approaches to actually develop classifiers of variants based on properties of the mutant sequences. And you know, this is something that we're going to be reporting soon. So you can imagine such an approach can, you know, you will get a cellular origin from plasma only NGS mutations. But clonal hematopoiesis is truly a very important confounder, if you will, in liquid biopsies right now.
Many institutions have molecular tumor boards?
Most large academic institutions do. And these are typically multidisciplinary teams, and these are expert opinion panels with expertise in cancer genomics, in clinical oncology, early clinical trials, genetic counselors, and so on and so forth. But they very much, to my knowledge, I mean, of course, you know, we have one at Hopkins that myself and Jessica Tao are leading, but these remain expert opinion based panels, right? And what that means is that you cannot scale up this effort, right, in terms of very carefully characterizing mutations by levels of actionability and then matching them with molecularly driven clinical trials. So we truly need what I described as a precision oncology decision support system. So in other words, you know, ways to automatically characterize mutations and match them with clinical trials or targeted therapies. And you can imagine that then this is important because it can occur outside of an academic institution, right? So we said that, you know, molecular tumor boards, they typically exist within large academic centers, but what about everyone else that is outside, right? It's a community. So that's why I think these efforts are particularly important.
Right. There are a couple more questions, and perhaps Dr. Anagnostou could stay on and either answer them in the chat or stay on and
sure, I'm happy to do
this.
Of course, happy either way. I'm happy to stay on and answer questions.
So this lecture was recorded and it will be posted where, Jill, if you would respond?
Sorry, I had to unmute. We usually post it on our website, and then FDA usually links to our website as well.
Okay, very good. So I want to take this time to thank my colleague, Dr. Anagnostou, for a very interesting talk about something I know very little about. So this was very, very good. All right, so thank you everyone for attending. Terrific attendance. Nice to see everyone. And if you would just stay on for a few minutes to address these questions, that would be great. All right. Good evening, everyone.
Thank you. All right, let's see.
Two questions at the bottom there. Best control for plasma-based cfDNA.
I see. Okay. All right. So
and the one right below it, if you would.
Okay. All right. So I'll get to it.
They may still be on if you want to speak out loud.
Oh, sure. Yeah, I can. I'm responding to the one whether liquid biopsies can be used to identify specific T lymphocytic expansion associated with tumor mutations. It may be actually better to to verbally respond.
Sure.
Yeah, no, I think this is an important question, and so we have some preliminary data, we and others, showing that there is. Together with ctDNA clearance, we see TCR clonotypic expansions, and so the idea there is that there is TCR T cell expansion of neoantigen specific clones. I would say, though, that I think overall as a field, the signal is not as clean as with ctDNA response or ctDNA molecular progression, just because T cell expansions, these occur in response to other antigens as well. And so a confounding signal that we at least have been seeing is the emergence of immune-related adverse events. And so if you ask me, these in terms of the clinical utility of looking at the T cell repertoire in the circulation, it is not as clear cut as looking at ctDNA dynamics. Another question has to do with differentiation of clonal hematopoiesis versus germline variants. Yeah, that's a great question too. And so this could be a little bit easier just because the germline variants in plasma NGS, these are typically around 50%, so very high levels, right? And then the variant allele frequency, or VAF, of clonal hematopoiesis mutations and tumor mutations, this is in kind of the 0.1 to 1% range. So one can, and again, all this is inference, right? But one can differentiate by levels in terms of clonal hematopoiesis versus germline. And then, of course, there is additional filters or filtering that has to do with whether an alteration is commonly seen, right, in germline databases and so on and so forth. Another question has to do with if certain liquid biopsy assays are able to accurately differentiate between somatic versus chip. Can this be done by liquid biopsy assays, or we would need additional clinical evaluations to confirm variants that may be clonal hematopoiesis related? So right now, there is no liquid biopsy assay that is plasma only that can be used to inform cellular origin, right? But what we typically do in reviewing liquid biopsies and seeing mutations, falling in DNMT3A or ATM or whatnot, or even P53, we tend to filter those out and focus on what we think is tumor specific, in terms, because what we're trying to do is match the tumor genotype with targeted therapies. But you're bringing up an important question, which is, should we follow up on these clonal hematopoiesis mutations? And there is more and more now, we recognize the importance of the association, obviously, with these chip mutations with myelodysplasia or myelodysplastic syndromes down the road. And so at least within large academic institutions, there's chip clinics or clonal hematopoiesis clinics that are now established, particularly for people, as we're doing more and more liquid biopsies, and we're detecting these mutations, there has to be follow-up, right? Especially with respect to down the road myelodysplasia. So this is now done, or could be done, in the context of specific, we call them, clonal hematopoiesis clinics.
Mm-hmm. Right, and I think the last question is Gregory Alexander's at the bottom.
At the very bottom, okay. Yeah, sure. So that's a great question, and this was just, I think, what I provided in terms of kind of the serial dilutions, if you will, of the genomic equivalence. This is just to emphasize the point that truly what we have in our hands in terms of mutant sequences and cell-free DNA is very, very low in levels. And so the point that I wanted to make there, and I hope that I came across, is the fact that the level of mutant DNA is very close to what is, sequencing error with conventional sequencing approaches. But certainly, so it was more so an example to highlight the scarcity of mutant sequences in cell-free DNA.
Right, I think we did it.
All right, perfect.
All right, very good. Thank you.
Thank you. It was a pleasure.
All right, wonderful. Bye. Thank you. Jill.
Bye. Thank you. Bye.